So today we are going to go over another of the 2025 FRQs for AP Bio. Today we are focusing on question 2. And remember, question 2 is one of the long questions, so it is recommended that you spend about 25 minutes on this question on the actual test. Go ahead and pause this part of the video so you can read the question, and then I will go ahead and go over it. All right, hopefully you paused, attempted to answer the question yourself, and then went ahead and restarted. So what we are asked to do is describe the polarity of a receptor that is embedded inside the membrane and the portion that's in the membrane. So if we remember, we've got our cell membrane. It's got a layer of phospholipids here, with the tail sticking in. And then it's got a facing layer of phospholipids with, again, the tail sticking on the inside. The important part to remember is that the head of a phospholipid is going to be hydrophilic, which means that it is polar. Whereas the tails of a phospholipid are going to be hydrophobic and therefore nonpolar. And we're asked about the section of the receptor inside the membrane, which means we are asked about the section of the receptor that is in this interior portion. And that is where the hydrophobic tails are. So that part of the receptor is nonpolar, because if it was polar, it would not be able to mix with the nonpolar sections. And you can also, if you want to be a little more specific, add that it's uncharged. Let's go ahead and look at the scoring guide. So some of the answers are that it is either hydrophobic or that it is nonpolar. Either of those would earn the point. Now we get into the meat of the question. So parts B through D are going to be referring to the idea of this 20E signaling molecule, whatever it is, and its relationship to the receptor dope ECR. Go ahead and pause so that you can read all of this, because I'm not going to read this out for you, and go ahead and read part B part 1 as well. All right, so hopefully you have restarted the video. Otherwise, I'm talking to myself. So we're looking at the description of an experiment. We're going to go over that a little more when we're asked about the actual experimental details. For now, let's focus on B, which is talking about constructing a graph. So question 2 always has a graph, and the graph is usually worth 3 points. So if you can graph, you can earn 3 out of 10 points without issue. This part of the test is also why the free response is not on computer, because you have to be able to graph by hand. So it's asking you to construct an appropriate type of graph. Notice we have these discrete categories. We have male moths injected with saline. We have male moths injected with an SI, or small interfering RNA. This means we're probably going to have a bar graph. When you have these discrete categories like this, it's a bar graph. If you have something that's a continuous form of measurement like time, then that's going to be more likely to be a line graph. It says your graph should be appropriately plotted and labeled as well. So there are 3 things that you are graphed on. One is labeling your axes. Another is having the right type of graph. So in this case, it would be a bar graph. And then the last thing is going to be having the data plotted completely or correctly. And then also notice we have error bars here. If you have plus or minus 2 SEX, that's plus or minus 2 standard errors of the mean. And that is going to mean you have to add error bars onto this. So in order to make this video a little shorter, I'm not going to graph this live. Instead, I am going to pause my recording while I do some of the graphing, so you don't have to sit there and watch me draw bars. But let's talk about what we're going to label this. So what we're going to label is, first of all, we're going to have two different groups. We have our control solution, and then we have our siRNA. So I'm just going to go ahead and put control. And with the siRNA for dope ECR. And then I'm going to put here that this is the treatment, because remember, it's good to have axes labels that also include units. And a good idea is to look at your look at your data table that they give you, because a lot of times that's going to give you your axes labels. And then notice that we have for our y-axis, what's measured is usually a % of time. So I'm going to go ahead and put % of time for my other axis, and % is my unit, so I don't have to do anything else. Now for the y-axis, I am going to have to numerically label it because I do have numbers there. I don't have to add any numerical labels on the x-axis. Remember, dry mix, if you're having trouble remembering what to put on what axis, your manipulated variable or your independent variable is on the x-axis, and your dependent variable or the responding variable is on the y-axis. All right, I'm going to go ahead and label the numbers, and I'm going to pause while I do that. All right, so I've gone ahead and I've labeled my axes. This isn't the actual graph section that was given on the test. I had to go and find it from another FRQ, but you'll just make sure that you fit the data. So our graph is going to go all the way up to 95 plus or minus 5. So the highest number would be 100%. And I want to fill up as much of the graph as I can. While I could label this where I had everything kind of squished into a smaller section of the graph, it's generally good to fill up larger chunks of the graph just because it makes it easier to see the data. So now I'm going to go ahead and I'm going to make markings where I'm going to draw my bars. So for the control solution with general activity, so I'm going to have two bars, general activity and oriented activity. It's 95%, which would be here. And there's my bar. This would be straighter if I was not drawing it on a computer. And then going over this with the error bars are going to be +5, so that's 5 above and 5 below. Now, they're not going to be as strict if the numbers are a little off. So these numbers are kind of multiples of 5. It's pretty easy to figure out where the error bars go. But make sure you don't have wildly off error bars. Like my error bars should not go all the way down to 80 or all the way down to 60. They give you a little bit of grace, but they don't give you all the grace in the world. Then the oriented activity is going to be 60% for the control group. And then notice I've got two different values, so I'm going to do some shading, and I'm going to include a key. So I'm going to say this is general activity, as % of total time. And I'm going to say that my slashed bars are oriented activity which is as % of general activity. To make sure that everything is clear and that I can very easily look at my data. Then my error bars are +4 and -4. And then I'm going to do the same thing for my siRNA. So that's going to be 90%. And the error bars are +8 and -8. And then the oriented activity is 25%. And that's ±6. So I have to have my error bars correct and I have to have my graph values correct in order to earn full points. Whenever they give you a plus or minus, that's always a sign that you're going to need to have error bars. And if you don't have those error bars, you risk losing a point. All right, let's go ahead and look at the scoring guide. So the scoring guide, each of these is a point. One is that it's a bar graph. That's 1 point. The second point is you have both the data and the error bars accurately plotted. So if you're off, then it's not going to give you that point. And then the last point is for an appropriately labeled graph. In the scoring guide, they do provide 2 examples. So the first example here, they both look very similar. One is activity of moths. Then they give you they give you the key underneath. Then they also say injected material, or let's see, here they're doing activity of moths and general versus oriented. So they, instead of graphing it by what is injected, here they graphed it by the type of activity. So either of those would earn the credit. Just note that you do have this extra key to tell you which bar represents what. So I ended up doing this by general activity versus oriented activity, but you could have also done this by saline injection versus siRNA injection. All right, now we need to look at the type of activity that was affected by inhibiting the expression. What type of activity was affected? Well, if we look, general activity was around the same. And if you want to see, notice there's overlap in the error bars. So that means that there was no difference. But here for the oriented activity, there was no overlap in the error bars. So that tells you that it's the oriented activity that was affected. And notice I don't restate the whole question. I just tell you which one was affected. Don't waste your time by restating questions. This is not an English exam. All right. Then go ahead and pause so you can read part 1. And now we have an identify. So this doesn't even necessarily have to be a complete sentence. We're trying to figure out where the oriented activity was greater than 50% of the general activity. So we look in this column, which one of those numbers is above 50%? It's going to be the control solution or the saline solution, because those are the same thing. That was the treatment group. If we look at the scoring guide, you could say it's the control group or the saline group, or you could say it's the male moths injected with the saline or control solution. All right, go ahead and definitely pause so you can look at all of this. This gets a little more complicated. All right, so now that you're back, notice now we're talking about a cell signaling pathway, and we're talking about a G protein coupled receptor. So we are saying that the scientists are going to mutate the G protein, so that way now we can no longer have the GTP bind. It's not going to displace the GTP bound protein, which means this pathway will no longer be active. So now we need to predict the effect of this mutation on the oriented activity. So notice it says when this pathway is actually working properly, there's increased expression of the genes associated with oriented activity. So this means when that 20E binds to the dope ECR, then it's going to increase the expression of genes with that oriented activity. When you block this, you're going to decrease the expression of those genes, which means you're not going to have the ability to orient to those pheromones. So this will decrease the oriented activity. And you're basically just predicting what happens when you don't have a receptor pathway or a signaling pathway working. All right. So if you look at the scoring guide, it shows decreased oriented activity. Go ahead and pause so that way you can read this explanation that we're going to use for part D. All right, hopefully you have read this and then restarted. So we're saying that the gene that encodes this receptor, and remember, you need the receptor for oriented activity. And they're saying it's not expressed when they are first sexually immature, when they first become adults. So then they're saying it increases as the moths reach sexual maturity. So they don't have the receptor they need for oriented activity. And then they build up the receptor that they need for oriented activity. And they're claiming this increases the likelihood of males finding females with whom to mate. Now, if we go back to the original paragraph, it's saying that the pheromones, the chemical signals released by females, are used to find mating partners, and that the orienting activity is them orienting towards their pheromone. So they're using this orienting activity to find females, to find mates. So we're saying that increase in gene expression increases the likelihood of males finding females to mate. So what we can say is the dope ECR receptor allows male moths to orient towards pheromones released by females in order to find a mate. Increased expression of dope ECR would, and remembering the information from the previous parts of the question for part C, would increase expression of orienting genes or orienting activity genes, which would increase likelihood, I think there's an E in there somewhere, of finding a mate. So if we check this on the scoring guide, there are a couple of different potential answers. So it's saying increased expression of dope ECR increases oriented activity and the ability to find a mate. Then another possibility is that increasing the dope ECR expression increases the chance of binding 20E to more receptors, or that increasing that expression makes the males more sensitive to pheromones from the females, or that it enables the binding of 20E to more receptors to make the males more sensitive to pheromones from the females. Now, notice my answers tend to be a little longer than these answers, but also I am trying to make sure that there's enough information there to earn the point. The answers on the scoring guides tend to be a little shorter because they're very short and to the point. Should your answers be this short? Not necessarily. It's more important to include all the information you need to earn the credit for the points for this test. All right, and the final part is saying, based on figure 1, explain how an inhibitor of this pathway would be basically a way to chemically protect crops from moth damage. So if you inhibit this pathway, you can say inhibiting dope ECR pathway would decrease expression of genes associated with oriented activity This would make it harder for males to find female moths to mate with. Because remember, this activation of this dopey CR receptor is going to increase the expression of those genes, and those genes make it easier for the males to follow the pheromones to find the females. So it would make it harder for males to find female moths to mate with. And it would decrease the moth population, because if they're not mating, they're not making babies, there are no more moths. And this would decrease crop damage. All right, let's go ahead and let's look if I can get this thing to go to the next slide. Hold on. Let's look at the scoring guide. So it says an inhibitor of the pathway would reduce any of these, the oriented activity or the expression of genes associated with oriented activity, and therefore decrease mating or decrease population growth. All right, so next up, I will do videos on the last couple of questions on 2025, and then if I have a chance, I'll go through 2024 questions as well.